Today's topic is about stem cells. We will use a handful of words that sound technical, but they are really not all that complex. It's like speaking about Peter, Paul and Mary. As I go, the words, we'll hear them, and in the course of the discussion I will explain what they are, what they mean. So what are stem cells? They are the building blocks of body parts. And there are over 200 types of stem cells in the human body. For example, there are skin cells, liver cells, kidney cells, you name it, it just goes on. There are brain cells, of course, and the brain cells are referred to as neurons. We'll be looking at that quite specifically today. And so with neurons, what do they do? Well, they do a lot of things, but in our case, we will restrict our discussion to those neurons that control body movement. They're called motor neurons. Relating to motor neurons, there's another technical word called dopamine. Dopamine is a chemical produced by neurons. One of its effects is to control body movement. So no dopamine, we're in trouble. And so we'll see how this pans out. Having said that, I actually want to read a bit from an article, a science article that I came across about a week or two. It's very, very significant, this. It discusses current research about stem cells and about how stem cells create neurons that produce dopamine. So I'll read out about 4 paragraphs from this article. It's very significant, and we will elaborate on what this article actually says, but I'll read this verbatim. So stay with me. You will understand this. In people afflicted with Parkinson's disease, neurons that produce dopamine begin to break down and die. The disease gradually produces tremors, involuntary movements, and trouble with walking, speaking, and other actions. While it currently can't be cured, studies are suggesting new ways to slow progression and reduce severity of the symptoms. An emerging and potentially groundbreaking treatment involves stem cells. Researchers have used stem cells to grow new dopamine-producing neurons, and then they transplanted them into animals. In so doing, they helped the dopamine neurons, the artificially produced ones, they helped restore brain circuits damaged by Parkinson's. The team started by converting human embryonic stem cells into dopamine-producing neurons in a laboratory. Then they transplanted these into the brains of mice, which had Parkinson's disease. Within a few months, the researchers saw that the new neurons had connected to the parts of the brain responsible for motor control, body movement, in other words. And the mice showed signs of improvement in their motor skills. Last para. A human clinical trial is currently underway in Japan. The 2-year observation period for that study is due to end soon. The researchers say that their technique could be adapted to other types of nerve cells as well, to treat different neurological disorders. So, in summary for this bit, stem cells create neurons that produce dopamine. And dopamine is a chemical that is needed for the brain to control movement. When neurons stop producing dopamine, people get Parkinson's disease. Scientists can create these new neurons using stem cell technology. We'll now have a look at a few slides. The first one coming up is slide 1. We see here a newly fertilised egg, one cell. At that stage it's called a zygote. This happens typically 16 to 20 hours after insemination. That's what we start off. So on the outside is the egg. It's been penetrated by sperm. For a little while, the two remain separate, although inside. Then phase 2, after 24 hours, the cell, the one single cell, divides into two, so that you can see in Fig 2 of our presentation here. So the egg remains the same size all the time, but the cells keep dividing, so they become smaller themselves. Now we'll go across to slide 2. Fig 3. We see now a 4-cell embryo. That happens typically after 45 hours. There are 4 cells. We'll just go one further here. In Fig 4, after 72 hours, the cell has now divided into 8 cells. And the process goes on, but we won't go down that path. What we have here is sufficient for what I want to explain. These cells, certainly at the 8-cell stage, and quite likely more, it's just that I've come across experiments where that included 8 cells. What scientists did, they'd remove the shell, take the 8 cells out because they're not yet connected to one another. So you now have 8 individual embryonic cells, they're called stem cells. The scientists then put an artificial shell around each of these embryonic cells. Then they implant, in this case, 8 separate cells into 8 separate wombs, and you have 8 babies being born. Had scientists not done this, had the first time when the egg was inseminated, when the sperm entered, you had one egg, initially one cell. If left alone in the womb, you'd get one baby being produced, untouched. That's the whole process, goes through clearly and cleanly. By doing what they did here, they showed that not only biologically that at this stage, these cells were not related, they were independent, but also we'll see that each of these little cells had its own little soul. In other words, the original one egg, one embryo had one soul. The soul is also, in the early stages, plastic. It can be pulled apart and you can produce many little souls. I might, as I'm speaking, it occurred to me that the church teaches that God only created one soul, that was Adam's soul. And so from Adam's soul and Adam's body, humanity appeared. And it's just, I remember, I think it was former Archbishop Paul, he gave the example where you have one flame and you light a candle from that flame, another candle from the flame. They all have flames. This flame does not get extinguished, but more flames appear. That's like the equivalent to the soul spreading from Adam. And so this shows, this particular process that we're discussing here shows that the soul, like the body, is malleable, and so it can be multiple. Okay, now typically these are called embryonic stem cells. They come from embryos that are 3 to 5 days old. So at this stage, the embryo is, another technical word, is called a blastocyst, and it has about 150 cells. Okay. For decades, all sorts of experiments have been conducted with embryos, including creating human-animal hybrids, human cows, human pigs, human mice, human rats, all sorts of stuff's been going on for, look, certainly 3 decades, probably for sure more, 4. Anyhow, and so in that scenario, after 14 days, these creatures would be destroyed. But we won't be discussing that at all beyond what I've just said. The thing that I'm aiming at to emphasise here is that the reason scientists are permitted to do these experimentations is the belief, the notion that the embryo is not a human being. At the very best, it's considered to be a potential human being. And because it's not a full human being, you can experiment with that. And this is what's been going on, and including in what we'll be covering now, the stem cell technology in this example to cure Parkinson's disease, but there are other diseases as well. Now, just to emphasise the fact that at no stage is from conception to birth, at no stage is there an actual border where you say that at this stage it's not a human embryo, but now it is. That doesn't happen. And I'll quote, I'll give a brief explanation, an extract from the book written by Lee Silver. That was back in 1999 from memory. He's a professor at Princeton University in the Department of Molecular Biology, Ecology and Evolutionary Biology, plus a thing called the Program of Neuroscience. So here we will look at slide 3, and I will read this, but I want you to read that with me, so I'll go through it slowly, and you'll see this slide as I'm speaking. So there's a couple of quotes from him. In the field of human reproduction, the word embryo no longer applies to cells that exist during the first 2 weeks after fertilisation. Instead, they're now called pre-embryos. Okay? So for the first 2 weeks, they're no longer called embryos, they're called pre-embryos, so even lesser human. I'll continue. These are quotes, these are words from Lee Silver. He says, I'll let you in on a secret. The term pre-embryo has been embraced by IVF practitioners for reasons that are political, not scientific. Why? To allay moral concerns that might be expressed by IVF patients. There is no particular part of the development process that is more important than another. All are part of a continuous process. From fertilisation to baby birth, nothing stands out as being specific and more important than something else. Last bit from Lee Silver. If we decide that the embryo is deserving of the same protection as a child, we might want to think carefully not only about abortion, but about kinds of embryonic manipulations as in the laboratory. Right, so to summarise so far this bit, we see that a single fertilised egg implanted in the womb produces 1 baby. Point 1. Next bit. After 3 days, the fertilised egg contains 8 embryonic cells. Third bit. These cells can be separated artificially and implemented, we discussed this, implemented into 8 wombs, thereby producing 8 babies. This confirms that each individual cell is now an embryo in its own right. Okay? And Lee Silver explains that there is no moment in time where the stem cells are not embryos. The embryos from word go to the end. Embryonic stem cells, they only have the ability to become individual embryos for a short period of time. Then they start to specialise. They start to connect to the surrounding cells, and then they start to specialise. And down the track, they cease to be embryonic cells. In the end, when full body parts are created, they are now called adult stem cells. All right. Okay, so here's a schematic diagram showing what happens. We start off in the middle there with those embryonic stem cells, each individual one, and left alone, they then start to specialise. It's called cell differentiation. So some of the cells become nerve cells, others become muscle cells, then others become heart cells, liver cells, and it goes on. There's just hundreds of them, different types. These nerve cells, muscle cells, and liver cells, they are now adult cells. So there's no problem doing experiments in the laboratory that use adult stem cells. The issue is when they are still in the early phase, are embryonic. Each embryonic stem cell, as I mentioned, is an embryo in its own right, and when the embryo is transformed chemically and artificially into body parts, that embryo is killed. And this is what we'll see. Have a look at slide 5. It's no different in principle to slide 4. In slide 4, right in the middle, we just showed the stem cells themselves. Now, this is no different to replacing that central image of stem cells with a fetus. And that's what I've got drawn here. So what's happening is that effectively we are transforming that fetus using technology to change from continuing to be born into a baby to change it, and it becomes an adult cell. So each time that happens, this little baby that you see there is killed in the process. Can you see where I'm getting to? So we started off with stem cells. It sounds, no big deal. They're just cells. But in fact, as Lee Silver pointed out, there is no moment when it becomes an embryo, when it becomes a fetus, when it becomes a baby. It's one continuous process. It's the same thing. And so if you destroy embryonic stem cells, you're destroying each embryonic stem cell is a little baby that is destroyed. So now we come to the interesting part. Prior to researching how many stem cells are used actually in these experiments that researchers do, and my gut feel was that, look, I felt maybe 10,000, even if you stretch it, maybe 1 million embryonic stem cells destroyed. And then eventually I came, I think it was yesterday, across some data, figures shown from the University of Utah. In this case, they were looking, experimenting with what are called blood marrow cells, but it doesn't really matter whether we're talking about blood marrow or any of the other cells. It's the process that's involved, and the figures that they speak about, they are indicative of the sort of things that we can expect in other areas of experimentation. So we will have a look at slide 6. This is an extract from the Utah University. This is what they say. the ideal number of stem cells needed is between 5 and 10 million per kilogram of the person's weight. That's the ideal number. The bare minimum number is between 1 and 2 million stem cells per kilogram of a person's weight. Now, the big deal here is per dose. So if you needed, say, 10 doses, you multiply this figure by 10. I don't know how many doses are involved. And so I've done this little teeny weeny bit of mathematics here. Say a person weighs 80 kilograms. And we'll look at the minimum number, where it's between 1 and 2. We'll look at 1 million. So if we multiply 1 million stem cells, embryonic ones, by 80 kilograms, we find that there will be 80 million embryos killed per dose. If we look at the ideal scenario where the university says the figure hopefully was between 5 and 10 million, if we take 10 million, we multiply 10 million stem cells by the weight of the person, 80 kilograms, and we get 800 million babies killed per dose. And if there are 10 doses, it's 8,000 million. That's a number of people on the planet. Do you understand why it's important to comprehend what on earth is going on? Why it's important for clergy in particular, but parents as well, and their subsequent children, for them to understand what's going on. Because this is, I'll give you now a scenario that you can expect in the Orthodox Church at least. It's very common for pious parishioners, when they have some serious issue that they have to attend to in their life, they will go up to the priest and say, "Father, bless me. I need an operation, or I need to do this, I need to do this, and it's very significant. I've lost my job, I can't find work. Can we do a little service to St. Xenia of Petersburg to help us?" And the priest says, "Well, of course." And the parishioner, all she's told, or he's told, is that we can cure your grandmother's or your mother's Parkinson's disease. We'll just use a bit of stem cell therapy. And because there's so many stem cells, that is, there are the embryonic, adult, but there's other ones in between as well. So the doctor's not going to say we're going to kill little embryos to fix up Parkinson's disease that your mother has. The doctor will say we'll just extract some eggs from the woman, we'll fertilise that with sperm, we'll produce stem cells, we'll then multiply these stem cells, and we will use that to cure, not cure even, to reduce the trembling and the consequences of Parkinson's disease. So to heal, or at least to help a person suffering from a disease, or other neurological diseases that were mentioned. I didn't just mention those, but that included even things like diabetes, for goodness sake. So to help one person to either get healed or helped medically, you can talk potentially 8,000 million embryos dying. During the Communist Revolution in Russia, over 70 years, typical figures that are quoted, that over the period of time, some 25 million people got killed. And that's, of course, horrific. And you mentioned it certainly in the church scenario to any priest or bishop or monk or seminarian, and they, of course, will all agree, horror shock, correctly. And yet they can be complicit in the death of hundreds of millions of embryos. Now, the church teaches that at Christ's second coming, when there's a resurrection of everybody, even these little babies that were aborted or miscarried, they got their own souls, and they will eventually have the appearance of a 33-year-old adult. So this is the essence of this lecture, to point out that it's not good enough for people, again, in particular my brother priests, to sort of say, well, science that's all very well, it's nice, it's out there, we can look at the trees and look at the beautiful flowers and smell the roses. And that certainly shows it's God's work. It's beyond that. We're talking about life and death. This is why knowledge about science is critical. And so if you're looking at young families, their toddler children don't need to know about science, but by golly, when they grow up into be mid-teenage, it starts to kick in. And especially when the world tells them that this is the new reality and they start to believe, then they'll fall away from their faith. And so it's time now for the church, I'm speaking in general, all Orthodox churches, and in fact indeed all Christian churches, to take this new world that we're living on board and understand we have to address issues that in the past were not even dreamt of. So that's the essence. This is the essence of today's talk, to show this is one example of why we need to know basic science.